So today we're going to be looking at a hormone called ABA. Sometimes I'll call it ABA. This is abscisic acid. It's produced by plants when they are under stress. So that might be because of a lack of water or perhaps just because of very low temperatures. It's involved in a whole range of different processes, including the control of stomatal closure when plants are short of water. And it got its name through its supposed involvement in leaf abscission. So that's when plants drop their leaves. However, it's thought that actually hormones auxin and ethene are perhaps more important in leaf abscission. Abscisic acid has been shown to cause leaf abscission in some plants, but actually it's also been shown to reduce it in others. It's thought that it might act by influencing the production and action of ethene on plant tissues. Now, I'll give you a moment just to have a little look at this graph. So as you can see, when the leaf water potential falls, both the stomatal resistance, i.e. how much they are closed, and the concentration of ABA increases. Then upon watering, both stomatal resistance and ABA levels decrease. Now, this could be a classic exam question. Not only would you have to describe a graph like that, the trends shown, but you might also need to explain what's going on. So here we have an image to help us understand how ABA acts. Now, ABA is a hormone, a plant hormone, that binds to receptors on guard cells. Now, when it does this, it causes a change in ionic concentration inside the cells. This then reduces the water potential inside the cells, as well as the turgor. So in this diagram, you can see on the left-hand side there, we've got hydrogen ions moving out and potassium ions moving in, water also osmosing in there. This causes guard cells to become turgid and we have an open stomata. By comparison, on the right-hand side, we've got ABA now entering in, as well as calcium ions. Chloride ions are now moving out. Similarly, potassium ions are as well. And water therefore follows by osmosis out of the stomata, causing the guard cell to go flaccid and the stomata to close. So this is the process by which the plant hormone ABA causes closure of stomata. Now, the second hormone we're going to be looking at today is a hormone called gibberellin, or GA for short. Gibberellin is responsible for causing rapid growth of stems. Now, an experiment, a simple experiment that can be done to just show this is by using dwarf beans. Now, dwarf beans are dwarf because they lack the gene for producing gibberellin. If you have a low concentration of gibberellin and you apply that to the stems of dwarf beans, the beans will begin to grow rapidly to the height of a non-dwarf variety. And these kind of experiments just show how gibberellin works and how it can cause that rapid growth of stems. Now, dwarf varieties of rice and wheat have been selectively bred that lack one or more of the enzymes needed for gibberellin production. What this means is that the dwarf varieties, being shorter in height, they end up putting more energy into the production of the grain itself rather than the growth of the stalk. This allows farmers to get far higher grain yields from some areas of their land. Gibberellins work by affecting gene expression. Gibberellin hormone moves through the plasma membrane and into a cell. Inside the cell, it then binds to a receptor protein, and that receptor protein will then bind to a selection of other proteins. This receptor complex ultimately breaks down a protein known as a DELA protein. DELA proteins usually inhibit transcription factors. They inhibit the transcription factors needed to transcribe a particular gene. Once that DELA protein is broken down, the transcription factor will be released and transcription of the gene can begin. And that transcription ultimately enables the growth of the plant, so you don't have the dwarf forms anymore. There's quite a lot on this slide, so I'm just going to give you a moment to sort of digest some of the information. On the left-hand side, you'll see a tall version of the plant, sort of wild type there, tall pea plant. And we're specifically looking at this one gene called the LE gene, or the LE gene. Now, the LE gene codes for a protein which is responsible for converting GA, gibberellin 20, to GA1, gibberellin 1. Now, if you have high concentrations of gibberellin 1, you're going to end up with greater growth. So this enzyme is really, really important. GA20 is essentially a precursor for gibberellin. Now, if you look over to the right-hand side, you'll be able to see the dwarf pea plant. Now, pea plants are dwarf if they don't have gibberellin, i.e. gibberellin is absent. This might be because you don't have a functional enzyme, so you might not have the LE gene as required. You might have a mutant form of it, so you might be homozygous recessive. Or it could be that you've got a lack of enzyme or one of these precursor molecules further up the chain. Now, a whole series of experiments have been done on this particular pathway, but specifically, one was done where there was a block between the production of gibberellin 12 adenylide, and this resulted in a dwarf plant. Now, what then happened is a graph was taken from a shoot of a non-dwarf plant, so a wild-type plant, onto this dwarf variety, homozygous recessive for the LE gene. And the resultant plant then grew taller. So there was more growth. Again, just showing how gibberellin acts to increase growth. Gibberellin or gibberellins are also responsible for other processes such as seed germination. So, for example, when a seed absorbs water, this activates the embryo, and the activated embryo will start to produce gibberellin. Gibberellin will then stimulate the production of different enzymes, such as amylase, which will ultimately break down food stores. So in the case of amylase, as hopefully you're already aware, that will break down starch into glucose. Now, that glucose will then be used by the plant for respiration, protein synthesis, and such like as it starts to grow. Now, this is a little bit of a tag on, perhaps, but it's yet another plant response that you need to be aware of. This is how plants respond to freezing temperatures. Now, if you have ice crystals forming inside cells, that's going to cause the cell membranes and the cell walls to burst, and that's going to kill the cells. So it's really important that plants are able to respond to such low temperatures. Now, they can do this in a number of ways. There's kind of 4 key ways that you need to be aware of. The first is through accumulation of solutes. So that's things like sucrose or polysaccharides, amino acids. So the cells can accumulate those solutes to reduce the freezing point and therefore sort of stabilizing the cell. Plants are also able to produce antifreeze proteins. These prevent the ice crystals from forming in the extracellular spaces. So those are the spaces outside the cells. The plant cells that make these antifreeze proteins tend to secrete them into the cell wall region. A third mechanism that plant cells can use to prevent damage as a result of freezing is that they can synthesize proteins called dehydrins. Dehydrins are inside the cell cytoplasm, and they bind to water molecules and alter the collective structure of water, stabilizing the membranes as a result. Finally, plant cells can alter lipid composition of cellular membranes in order to adjust the fluidity to cold temperatures.